Every human body begins as a blank slate of stem cells. During development, these cells travel down a one-way biological timeline, adopting rigid shapes to become muscle fibers, skin, or neurons. Biologists call this permanent specialization cellular commitment. This specialization creates a vulnerability. Because locked cells have lost the ability to revert to a blank state, they inevitably accumulate damage and wear out. This physical decline is the biological basis of aging. For almost every multicellular organism on Earth, this one-way street ensures that death is an inescapable physical consequence of having a complex body. Some species have evolved unique survival workarounds. The freshwater hydra escapes aging by continuously replacing its entire body, swapping out every cell every few weeks using a permanent reservoir of stem cells. The tiny Mediterranean jellyfish Turritopsis dohrnii operates outside these boundaries. It possesses the ability to force the timeline of cellular commitment to run backward. When threatened by starvation or injury, the fully grown adult medusa reverses its life cycle. It retracts its tentacles, collapses its bell, and deconstructs its body until it settles on the seafloor as a juvenile polyp. Deconstructing a complex body is a high-risk mechanical maneuver. To survive the transition, the jellyfish must melt down its tissues without destroying the genetic blueprints required to build itself again. The reversal begins with a cellular alarm. Physical trauma or starvation triggers a sudden influx of calcium ions, which signals the organism to begin the teardown. Genetic programs activate to dismantle the medusa's specialized structures. The centralized nerve ring and striated muscle fibers dissolve, reducing the complex animal to an amorphous cyst on the ocean floor. Dissolving organized tissue exposes the genome to immense physical stress, leaving the DNA vulnerable to mutation and terminal fragmentation. The jellyfish counters this risk by activating POT1 genes. These genes synthesize proteins that encapsulate the telomeres, the protective caps on the ends of chromosomes, physically shielding the DNA strands while the rest of the body liquefies. This strategy treats the physical body as expendable. The jellyfish sacrifices its specialized tissues to ensure the absolute protection of its core genetic code. Once the DNA is secure, surviving cells undergo transdifferentiation. Every specialized marker is wiped, forcing cells back to a blank multipotent state. They then adopt new instructions, re-specializing into a completely different role required for a juvenile polyp. The pace of the rebuild is highly efficient. In laboratory conditions, safe mitosis resumes within 24 to 72 hours of the initial collapse. The amorphous cyst sprouts into a healthy juvenile polyp. This new colony is functionally immortal, budding off identical medusa clones to restart the cycle. This is rejuvenation via total replacement. The organism generates a young body by systematically reprogramming the materials of the old one. On a cellular level, Turritopsis has mastered immortality, but it remains physically fragile. In the open ocean, these jellyfish are consumed by predators, crushed by currents, and killed by disease every day. Human shipping accidentally turned this fragile survival mechanism into a global advantage. When jellyfish are sucked into the stressful, dark environment of a ship's ballast tank, they initiate the teardown. They survive oceanic voyages as protective cysts and emerge as polyps once the ship reaches a new port. This ability to survive global transit through life cycle reversal has transformed a localized Mediterranean species into a successful, worldwide invasive organism. Gerontologists study Turritopsis to identify the precise pathways that allow complex cells to reprogram themselves without triggering terminal errors. In humans, when cells lose their specialized identities, they often grow uncontrollably, resulting in cancer. The jellyfish manages this same cellular reset while maintaining perfect biological organization. Mastering transdifferentiation would offer a direct method for engineering human tissue and advancing the capabilities of regenerative medicine. Turritopsis dohrnii proves that cellular specialization is a reversible state, providing a biological precedent for starting a life cycle again using existing tissue.